right. Well, welcome to the fifth and last seminar of our Iowa Nutrient Research Center seminar series for spring 2021. I'm Matt Helmers at Iowa State University and the director of the Iowa Nutrient Research Center. It's been really enjoyable this semester to have some speakers from across the country to be able to talk about some of their work looking at nutrient loss on a watershed scale. And we've kind of traveled from Arkansas to Nebraska to Ohio, and then Jason Hubbard that talked a little bit. He kind of traveled all over the country because he talked a little bit about in the West and his work in Missouri and West Virginia. And now we are very fortunate to kind of come to one of our I states and have Dr. Jennifer Tank at Notre Dame University. And Jennifer does a lot of work on nutrient loss, and I bet a lot of us know her most for some two-stage ditch work, but also watershed work and I think some watershed work with cover crops. So we are really excited. I think all of us would love to be here in person, but for everybody's travel and for our own travel budget, it's nice that we can do these things virtually. So I'm going to turn it over to Dr. Jennifer Tank. When we get to the end, if you have questions, or even as we go, if you have questions that come up, feel free to type them in the chat box. We'll also allow folks to unmute themselves at the end if they want to ask questions verbally rather than just chatting in. We welcome that as well. So with that, thanks again for attending, to all the attendees, and thank you, Dr. Tank, for being with us today. And now the virtual floor is all yours.
Great, thanks, Matt. I really appreciate the invitation. Let me see if I can share the screen and get us into full screen mode. Is that working okay?
Yep, it looks great on my end.
Okay, fantastic. Thank you. So yeah, a pleasure to be here, and wonderful that your theme is watersheds. At least I have watershed in the title, which is great. Today I'm going to talk to you about linking soil health to improve water quality via the planting of cover crops, using example from 2 Indiana watersheds that we've been working in for a number of years. My partner in crime is Todd Royer at Indiana University, and we wanted to especially acknowledge our students. Shannon, Matt, and Leanne have been working for a number of years on this project. Shannon just got her PhD on I got to hood her on Saturday, so it's Dr. Spear to me now. And Dr. Trentman is now a postdoc out in Montana. So we've had some great students on the project, wanted to acknowledge their contributions. So I'm sure I don't need to tell this group that waterways draining row crop agriculture reflect leaky systems. We know that hydrologic alteration is necessary for productive agriculture, channelization and subsurface tile drains, speed water removal from fields. But excess fertilizer, nitrate, and soluble reactive phosphorus are also transported from fields to streams and ditches. And highest losses occur after harvest during the fallow period when fields are bare. And so this decrease in water residence time, a decrease in nutrient retention, really results in this increased nutrient export that we hear quite a lot about, and it drives our need for conservation that limits nutrient loss, particularly during winter and spring months from agricultural lands. And we've all seen the poster child for why we should care about this. The export of excess nutrients contributes to algal blooms, followed by hypoxia. The Gulf of Mexico is the classic case. This figure from NOAA that came out a few years ago, so powerful. It does vary from year to year, and it really depends on the water year, but we're not quite to reaching our goal of reducing nutrient export from the agricultural Midwest. I'm a little bit closer in Indiana to Western Lake Erie Basin, where algal blooms are really high profile, driven mostly by delivery of dissolved phosphorus. We call it soluble reactive phosphorus in sort of my area of biogeochemistry. It's the same as DRP, dissolved reactive phosphorus, that you hear talked about with regard to Western Lake Erie Basin. But it's really driven by April delivery from the landscape to Lake Erie. And those blooms are highly correlated with that runoff. And every few years, we get these very large algal blooms that make the national news. This picture in the lower left is from the Toledo Blade in September 2017. And then in really big years, they start to talk about very big changes, like giving natural waterways like Lake Erie the same legal rights as people. So really something that if agriculture can get ahead of this, we're all better off. And what the future holds is probably even more challenging. In Indiana, we predict a large increase in 100-year storm events by the 2080s. That means that more water is going to be pushed out of the system with these extreme events. And this happened in February 2018 in my home watershed, which is the St. Joseph River Basin in Indiana. That February 2018 event was a 2,500-year flood event. And these multi-100-year events, even to 1,000-year events, are getting more and more common. And I'm going to show you the influence on some of our watersheds of that February event. But really, what I'm trying to drive home here is that the challenge for us is how we can maintain productive agriculture, maintain drainage, while preventing nutrient and sediment loss to adjacent waterways, especially during these high flows. So the practice I'm going to talk to you today about, and I'm sure again this crowd is well versed in this, but I'll do my spiel anyway, are cover crops. They provide this vegetative cover outside the cash crop growing season. I show an example from one of our watersheds that we work in, the Kirkpatrick Ditch watershed, where a traditionally managed field is abutted right against a cover crop field. And we know that cover crops increase evapotranspiration, take up nutrients, increase soil organic matter on a little bit longer time scale, and really that the magic with cover crops is happening below ground. So this goal that drives our research to prevent nutrient runoff to adjacent waterways, cover crops is one of the ways, the watershed scale approaches we're exploring. In the Midwest, it's gaining, or sort of maybe peaked in gaining popularity, but it's still less than 10% of croppable acres in the Corn Belt. Indiana was doing great there for a number of years, and now we've sort of plateaued out just below 10%. There's been a lot of excellent work on the impact of cover crops in terms of edge of field studies. Very consistent reductions in tile drain nitrate losses, but variable results for soluble reactive or dissolved reactive phosphorus. So there's still a bit, the jury's still out on that in some ways. Modeling studies, mostly or many using the SWOT model, suggest that export reductions at the watershed scale could be from 20 to 67%. So these are the types of numbers we want to see. But there have been very few published studies quantifying the impact of cover crops on working lands. But there are a few. They're mostly focused on nitrate reductions. Those reductions range from 13 to 34%. But I think there are some critical gaps that we've tried to address with our work. One is linking the field and watershed scale effects of cover crops. And second, quantifying the impacts for both nitrogen and dissolved phosphorus loss on these working lands. So today I'm going to talk to you about the Indiana Watershed Initiative. This project was funded by a USDA Regional Conservation Partnership Project, RCPP program. Many of you are probably familiar with that. There's most of the money that comes through an RCPP grant is for the implementation, the incentives for implementation of those conservation practices. There's a little bit of technical assistance money as well, and we had a 4-year project. And the idea was to look at how the planting of cover crops could improve soil health, decrease nutrient loss from tiles to waterways, and then document the watershed scale effects of improved water quality. So today I'm going to go through those 3 sort of areas. I'm going to use this little symbol for soils, the symbol for tile drain data, and finally the waterway data. And so the idea is that we want to keep leaky nutrients on the fields during winter and spring. So let me tell you this tale of two watersheds that is the EWE project. Just for a little context, Indiana is about 85% row crop agriculture in corn and soybean rotation. The Chateau Ditch watershed, which is sort of centrally, north central Indiana, is about 3,200 acres, mostly no-till agriculture. The Kirkpatrick Ditch watershed is about double the size, 6,400 acres, and more traditionally cropped acres, and more rented ground than owned ground in Kirkpatrick. The soils are fairly similar in those two watersheds, mostly fine loamy soils, but there's a little more clay in Kirkpatrick Ditch watershed. The inputs, the practices by individual landowners are similar in that they have spring starter with the summer side dress, but Kirkpatrick Ditch watershed has some fall manure application where Chateau does not. Today I'm going to show you data for the water data from the tile drains and the waterway, 4 water years of data starting in 2016. I'm going to start though with some soils data, and that started a little bit earlier. We've been working in the Chateau Ditch watershed actually since fall of 2013. But just to give you an overview of the sort of implementation of the project though, the cover crop coverage as percent croppable acres in the Chateau Ditch watershed ranged from 62 to 68% of croppable acres, whereas in the Kirkpatrick, we went from about 13 to 32% over that 4-year implementation period. And just to give a perspective, I need one because I can't really think in acres, but at Notre Dame, we can think in football fields. It's about 3,000 acres planted per year in both those watersheds. So via the project, about 2,300 football fields a year. So how did we quantify these potential benefits of cover crops? Well, for the soils, we sampled 2 times per year, once in the fall post cash crop harvest, and once in the spring pre-cover crop termination. And so what we're going to be comparing, I'll show you data from means from cover crop fields versus no cover crop fields. And we analyze them for soil nitrate, water extractable phosphorus, and percent organic matter is what I'll show you today. And then for the water, we did use sort of a synoptic sampling approach every 14 days for 4 years. So it's a total of about 36 tile drain outlets in both watersheds. It's about 2 per field, and about 13 longitudinal stream sites going from the top of the watershed to the bottom of the watershed in each. Then sampling at both watershed outlets. And I'm going to show you data today for nitrate and soluble reactive phosphorus that we analyzed on the Latchet flow injection analyzer in my lab. The data represent about 3,000 measurements over those 4 years. And then finally, flow discharge is really critical for any of these measurements because we want to express things as mass loss rather than concentration. So for the tile drains, we have instantaneous discharge at the time that we sample each of those tile drains. So it's basically with a bucket and a stopwatch. And so that represents sort of instantaneous Q. And then at the outlets, we have USGS gauges for continuous discharge. And these are pictures of the students sampling in the field. So let's start by setting the stage for the cover crop, potential cover crop reductions by looking at what happens to cover crop biomass. And I just wanted to remind, so this the orientation of these soils figures, the left will always be Chateau Ditch watershed, shaped kind of like Italy, really. And it starts, if you can see my pointer, it starts here and moves to the outlet here into the Tippecanoe River. And this is a depiction at 67% coverage. And then the Kirkpatrick, the bigger watershed, this happens to be one of the middle years where we had 25% coverage, is here on the right. And I just wanted to remind that this was, we basically, in partnership with the soil and water conservation districts in those watersheds, we paid the farmers to plant cover crops at $45 an acre. We did not tell them what to plant, and we did not speak about inputs or any other management that they were doing, just the incentive to plant cover crops. And so what got planted sort of varied from year to year and field to field, and frankly, watershed to watershed. They mostly coalesced independently, just talking to each other, on either annual or cereal rye was the most common planting, but then there were some mixes that were explored by individual farmers, etc. So I'm just going to show you generally what that cover crop biomass looked like, and this is above ground biomass. And really what I want you to see is that these are, again, working lands where we're not controlling what's happening. And biomass generally increases from fall to spring sampling, which is great and what we would expect. So we've got more biomass on the ground in spring, which is good for potentially mitigating some of those losses to waterways. But things vary annually. Some years were just really great years for cover crop growth, others not so much. But it also depends on rotation and field scale variation, individual producers, etc. So I just wanted to give you that context. But surprisingly, some of the soil nutrient data are pretty consistent despite this variation. So let me show you soil nitrate, which is our leaky solute first. And again, shadow on the left, Kirk on the right. And what you can see is that both watersheds early in the study, and I'm showing you 0 to 5 centimeter data. We also have 5 to 20, but the patterns were very similar. Soil nitrate was consistently lower in fields with cover crops, especially early in the study, but trends really varied annually. And you can see in particularly wet years like water year 2018 and 2019 in the Chateau ditch, there weren't significant reductions. So the long-term data set that we have really helped us sort of see this interannual variation. For water extractable phosphorus, it was a little different between the two watersheds, not as consistent. What I do want you to note is on the y-axis, the water extractable phosphorus pools were generally a lot larger than nitrate by about 10-fold. So there's a lot more phosphorus in these soils. In the Chateau ditch watershed on the left, water extractable phosphorus was consistently about 20% lower in fields with cover crops. But we did not see that pattern in the Kirkpatrick ditch watershed. We really didn't see consistent reductions, and there was sort of a flip-flopping back and forth with cover crops. And the soil setting, aluminum and iron are the most different in these two watersheds when we sort of average across all the fields that we sampled. That's one potential factor that could be influencing the difference in the impact from Chateau to Kirk, as well as the fact that they are periodically adding manure in fall in Kirkpatrick, some fields, some years. Remember, these are means of all the no cover crop fields versus the cover crop fields. So it's interesting that the one thing we're not seeing is a huge increase in loss of water extractable phosphorus with cover crops. That we did not see. And finally, what the producers really care about is what we saw in terms of percent organic matter. And that, when we pool the data among the fields, we did not see significant difference between fields with and without cover crops over the study period. The Chateau ditch, if you look at the green dots as they move through time, you see sort of this gradually, and I'll use my pointer here if I can. There it is. You can see this sort of generalized increase, but that trend was not present in the Kirkpatrick ditch watershed. It's just sort of straight across the board. And so what we think we really need to explore is field by field by field for temporal trends, and that's really a next step. These are the pooled data. And it could also be that the time, the lag, it's just not long enough of a study to pick up the soil organic matter changes. That soil organic matter pool, when you're talking about 3%, 4%, 5% organic matter, that's a lot of organic matter to change. That pool is hard to change. Okay, so that sort of sets the stage. Potentially good news in the soils. Let's move now to the nutrient loss rates via the tile drain. So this is, I call it's edge of field sampling. We are sampling at the edge of field, but it's not, it's sort of EWE style, if you will. We're focusing on getting a lot of spatial and temporal coverage here in doing it by hand every 2 weeks. We don't have automated samplers to do it at single places in real time. So what I wanted to make sure to point out is that we are taking our nutrient concentration data from each of the water samples we sample at each tile drain and multiplying it by the instantaneous flow to get a mass loss and just extrapolating that up to the day. So I'll be expressing these in kilograms per day. Okay, so how do cover crops influence tile drain losses? So these tile drains, I'm pooling the data now. So it's tile drain data, you can see from the little symbol on the left, for both shadow and current combined, all those tile drains together. And what you see, brown is no cover cropped fields, green are cover crop fields. That cover crops consistently reduce tile drain discharge or flow, which has implications for the nutrients that are coming along with that water. Cover crop tiles were 28, depending on the year, notice it's 2016 through 2019 water year, depending on the year, 28 to 65% lower flow than no cover cropped tiles. We think that cover crops are increasing the water holding capacity of the soils via that root biomass and the intact sort of soil column, as well as the potential for increasing evapotranspiration by the plant itself. Let's look now in the middle, the middle panel at reductions in nitrate loss. They're similar to our magnitude of reduction in flow. And so they sort of mirror each other here. So flow really, this leaky solute of nitrate, flow is really driving that nitrate loss. The reductions range from 27 to 72%. They were significantly lower in 3 out of 4 years. Water year 2019, there was a trend, but no significant difference between the tile drains. And then finally, on the bottom row panel, you can see that the data for SRP. So cover crops also reduced SRP losses, but again, a little bit more variable in terms of the range of reductions from year to year. So 7 to 59% reduction, 3 out of 4 of the years were significant. Again, water year 2019 was not significant. But we think that the differences in the field scale losses are most strongly influenced by management choices, other management choices besides cover crop planting, in combination with differences in the soil properties among those different fields. And one thing I wanted to show you is that we really wanted to try and understand, it's a lot of tile drain data, 30 tile drains every 2 weeks for 4 years. It's a lot to get your head around, and Shannon did a fabulous job doing that. What we wanted to understand is, how are these tile losses distributed over time and space? And what we found is that they're variable. So I wanted to give you sort of a perspective of what I'm showing you here. This is the top of the watershed to the bottom of the watershed. Here's the watershed outlet. We've got the 4 water years, and then here are the tile drains, the individual tile drains moving from top to bottom of the watershed. We've got flow data in blue, nitrate mass loss in green, and then SRP mass loss in oranges. And the darker the color, the higher the value. The C's represent tile drains that drain cover cropped fields. And so what you see is tile drain losses are really spatially and temporally heterogeneous. And we saw the similar pattern in Kirkpatrick. So the left 3 columns are Chateau and the right 3 columns are Kirkpatrick Ditch. And that was really surprising that heterogeneity came through in both watersheds. But the biggest surprise to us was that these hot spots and hot moments in these watersheds for nitrogen and phosphorus seem to mirror each other. We weren't expecting that given what we know about the biogeochemistry of nitrate, which is really leaky, and what we think about with phosphorus as being sticky. Flow seems to override that pattern, and we see this mirroring of nitrate and SRP loss that's driven by flow. And so just another visual I wanted to give you is how these, now that I've showed you that tile drains are spatially variable as well as over time in these watersheds, I wanted to show you how that influence what you see in the waterway as you move from the top to the bottom of the stream. So how do tile drain losses alter longitudinal stream loads? So it's sort of combining, as you can see in the left-hand corner, the tile drain data with the waterway data. And what we generally saw in the upper left-hand and right-hand corners for both watersheds is that sub-watershed nitrate loads, at each longitudinal sampling site, capturing everything from moving upstream from that sampling site, we call a sub-watershed. Each of those, as we move downstream for sub-watersheds, sub-watershed nitrate load really increased with flow. And the inset diagram here, let me find my pointer right here, the insets for both of these watersheds show as you go from the top of the watershed in the cool colors to the bottom of the watershed, which is the outlet, you get more and more. So it's leaky and it's sort of, it's accumulating as you move downstream, and it's highest at the outlet. What was fascinating is that we didn't see that pattern at all for SRP. Instead, SRP loads didn't really follow discharge in the way that nitrate did. Instead, the highest loads sort of bounced around in the watershed. They varied over time and over space. So the y-axis, the x-axis on the insets is time. So they kind of blinked on and off in the watershed. We think they were event driven. So in terms of how cover crops could be implemented to reduce nutrient loss, widespread planting of cover crops would be needed to reduce nitrate losses because the losses are diffuse in the watershed, moving from upstream to downstream. In contrast, SRP losses appear to be patchy, and perhaps targeted implementation could do the job. But I think if you want to do both, we have to err on the side of scaling up to watershed implementation. The other thing I kind of alluded to with all of this is the role of events, of storms linked to precipitation, and how that might influence what we're seeing in terms of tile losses. And this is from Matt's dissertation work, looking at how antecedent precipitation influenced tile drains. And what we found is that when we look at storm-related phosphorus loss from these tile drains at both the watersheds, cover crops are holding back water, and hence with that water holding back the SRP to minimize that export. So if we look at the difference between the cover cropped tile drains versus no cover crop tile drains, both for flow and for SRP loss rates, what we see is that the antecedent precipitation, 7-day totals, really influence how much is lost. These are on a log scale, influence how much is lost, but that generally the whole line for cover cropped tile drains is lower. So it sort of pulls everything down relative to no cover crop. So yes, precipitation really matters at sort of that 7-day total. It is the most significant driver of what we see in terms of SRP loss, but cover crops reduce that overall loss by sort of an offset lower. Okay, finally, I guess what the Gulf of Mexico or Western Lake Erie Basin residents care about is what ends up being exported downstream via the watershed outlet. So I want to set the stage of how we went about addressing that by looking at our watershed export. So remember, we've been doing nitrate and SRP grab samples, and we have continuous discharge at the outlet. This is what our data set looks like. Again, we started in the Chateau Ditch watershed much earlier, and then added with the RCPP funding, we added in the Kirkpatrick Ditch, which is the second row. But what you can see from the system is that it's flashy. That's the blue lines, precipitation across the top in the black. So this is very responsive. And then all the dots in green and in orange represent those grab samples. And we took this multi-year record and modeled watershed nitrogen and phosphorus export using load flex, Allison Appling's program. So what I'm going to show you is the impact of cover crops on spring export of nitrogen and phosphorus. So April to June period, which is the period when cover crop biomass is the highest and when we would expect the reductions, and the time of the year that really influences those downstream algal blooms like we showed for Western Lake Erie Basin. So first, and now we've put Shadow on the left, Kirkpatrick on the right, and these are expressed as yields, so kilograms per hectare. So now the watersheds are on even ground here. And what I want you to notice is that the scales are different. Nitrate yield top scale going to 20 kilograms per hectare in Chateau and 40 in the Kirkpatrick. But in 5 out of 8 site years, so we have the pre-data, the pre-cover crop incentive data is a little bit longer for Chateau because we were longer in that watershed. Only 1 year for Kirk, so it's not the perfect experimental design, but then we have 4 watersheds in each of the 4 years in each of the 2 watersheds. So for 5 out of 8 site years, we saw reductions in spring nitrate yield from the watershed. And if we average, when we did see a reduction, it was about 36%. But there were years when we didn't see reductions. And that's where the multi-year monitoring has really come into play, because what comes off of the watershed very much reflects the runoff. So this interannual variability in runoff sort of overrides the signature of conservation, and it's very predominant. And what we see is in the 2 years where in Chateau, for example, where we saw higher than the pre-data, higher exports in spring, were also years that had 50 and 140% more runoff relative to the pre-year. So this overriding signature of the water year is a challenge. For SRP, again, notice the difference now in the case Chateau is about double the scale of Kirkpatrick. So Chateau is losing more phosphorus than the Kirkpatrick. But in 7 out of 8 site years, we saw reductions in spring export. And the average of those reductions was about 51%. What was really surprising to us, I think most, was that the percent reductions were similar. I mean, they're not exactly the same, but they're not out of whack from each other, even though the variability in the watershed scale coverage of cover crops was different. Remember, we got up to 67% coverage in the Chateau, and only the highest was 35% in the Kirkpatrick. So despite the differences in environmental context, like soil type, input rates, individual field management, cover crops did appear to reduce, on the majority of years, reduce nutrient export in spring, which really, I think, is good news. It could be critical given the predictions for increased storms that are forecasted for the Midwest. So I just want to show how influential storms are in these data to sort of close here. Here is a flow duration, so flow duration analysis where we've got our different flow categories. So the top 10% of flow events, the 90 to 100th percentile, are the lightest colors, and how much of the annual export each year is responsible via the top 10%. So what I want to emphasize to you is that the top 10% of flows, really, really important in terms of driving nutrient loss. So 40 to 68% of annual nitrate export left in the top 10% of flows over the 8 watershed years of our data set. Even more dramatic for SRP storms, these event-driven losses are even more important. So 48 to 93% of annual SRP export was driven by the top 10% of flows. And I put red boxes around that February 2018 storm. More than 90%, so about 93% of annual export happened on one storm event. So, you know, wiped out basically all the good done by conservation, one extreme event like that. And I really think this is an issue that needs to be discussed, because these future climate predictions are really going to make what's already challenging, which is to document watershed scale success with conservation. It makes it the hill even higher to climb, given what's predicted for future climate. So for agricultural watersheds, anything that enhances the resilience of these systems, makes them more like sponges in the context of these high flows, is going to be absolutely necessary. And I think it's particularly a challenge for SRP, where we see more event-driven, isolated event-driven runoff that drives losses. Okay, so I've thrown a lot of data at you. I just wanted to give you some overall take-homes going from bare soil, fall to spring, to this land cover change in winter cover crops. For soil health, we saw, especially early on in the study, about a 60% reduction in soil nitrate. That changed in the later years that were wetter water years, about a 20% reduction in soil water extractable phosphorus. For tile drains, the reductions in losses from fields were variable through the 4 water years, but range from 27 to 72% for reductions in nitrate loss rates, and 7 to 59% for SRP. So SRP is a little bit more variable than nitrate, and we think it's due to those events. And finally, in the context of watershed export, looking at spring nitrate loss, 5 out of 8 site years, we saw reductions, and that average reduction was about 36%. For SRP, 7 out of 8 site years, and the average was a 51% reduction. So these are magnitudes that are, I think, important, especially given, I mean, these percentages are from watersheds that export a lot of nutrients. So the potential is there for watershed scale cover crops to do the work, but storms really drive nutrient loss, and we don't have a lot of control over those storms that are coming down the pipe in future. So finally, as a take-home, watershed scale cover crops can keep nutrients on fields and have the potential to reduce watershed losses. I just wanted to add a few comments about scale. Sorry, Matt, I was finally, but not finally. I just wanted to mention that we've measured things at a lot of different scales with this study, and I think we've learned some things about it. At the field scale, the multiple tile drains that we've sampled, we did see this consistent reduced tile mass loss for both nitrate and SRP. But one of the really key features was that we did year-round sampling and multi-year sampling. We could have missed this depending on when and how long we measured it. At the sub-watershed scale, I'm glad we did that longitudinal sampling because we saw some spatial heterogeneity in loading, and that highlights the need for, if we want to get it all, we really have to saturate the landscape with cover crops in order to achieve the really kind of massive reductions that are being called for by federal agencies. At the watershed scale, I think there's potential that cover crops can buffer storm-driven nutrient losses, especially in spring, but we're going to have to do it at scale. And I think the argument that talking about resilience in the context of management conversations, in addition to the soil health benefits that we already discussed with producers, could pave the way for more cover crop adoption at scale. And so what I've shown you today is, I think my students have really benefited from the experience of working in these agro ecosystems. They've learned the value of long-term partnerships with the Soil and Water Conservation District, the NRCS, the producers. They've really been partners in terms of the co-production of knowledge and how the work gets effectively translated. So decision-making changes. And I think that they are learning a lot of skills. So I encourage folks that are training their students for the challenges of working on working lands are, I think they're there definitely, but the benefits far outweigh that. These partnerships, we especially thank the farmers, the Soil and Water Conservation District, the Nature Conservancy was a great partner for us, and the USGS. And then, of course, none of this would have been possible without the funding. The RCPP program required match, and the Walton Family Foundation and Indiana Soybean Alliance really came through with the match money. So we're very grateful for that. And now, Matt, I am really done. So I'd be happy to answer any questions if there's time.
All right. Yes, thank you, Jennifer. That was great. And we have some questions. I'm going to take the prerogative as the moderator to ask the first question, my own question. So on the one slide, you showed the flow reduction, I think from the tile lines, you had the flow reductions with cover crops, and then you had the nitrate reductions, and they were really similar reductions. And so that indicates to me maybe you didn't see much difference in the concentration of nitrate between the cover crop and non-cover crop field.
Right. Yeah, and one way we haven't, because I can't get my head around it, we haven't looked at, but we could look at is to look at flow weighted concentrations. That might be another way to relay the changes. But the changes appear to be flow driven, and the nitrates just coming along with the water.
Interesting. Yeah. Yep, and kind of related to some of that that we questioned from now. I'll say this is a question coming from others, so I'm not just taking all the time here, but kind of related to, I think, some of that drainage data. Were you able to quantify or control for the drainage area contributing to those tiles when compared?
Yeah, no. So we don't, I mean, even the landowners don't have it. There were a few tile drains where they were put in very recently, so they have the tile maps. That's why we tried to cover the whole watershed with the tile sampling, to say that at least we know we're spread across the watershed and we're hopefully capturing similar areas. But you're exactly right. I think given that imperfection that we had, to see the consistent reductions relative cover crop versus what we call cover crop field tile versus no cover crop is pretty amazing, given the fact that we don't actually have those tile maps and we don't control for tile area. So you're exactly right on that. Yes.
And then kind of related to the panel of three from the flow and the nitrate and then the soluble reactive phosphorus. The question was, it appears that the difference in SRP between the cover crop and no cover crop was less than that of Q. If correct, it seemed to suggest an increase in concentration of SRP under the cover crops.
Yep, that's a great point, and that could be so. Remember, I mentioned the manure application that's still occurring some falls in the Kirkpatrick, and I should have given the context in Chateau, the historic sort of legacy phosphorus in Chateau. These are both watersheds where the background soil test phosphorus is high. They put chicken litter on in shadow for many years before they stopped. And so the point is there's a lot of phosphorus to lose in these systems. And I think that may be where phosphorus doesn't follow the water exactly. So maybe, yeah, I think looking at flow weighted also showing these data as flow weighted concentrations could give a little bit more insight that the audience is picking up, which is great.
Yeah. And there was a question whether you looked at total phosphorus at all, even at the kind of watershed outlets?
We do not. We do not. We have a lot more. We have every which way you can measure the soil phosphorus, but due to sort of logistic constraints and cost, I don't. Those digestions are would have to be sent out rather than doing them in the lab, and it was just something we couldn't afford. And I know our data without the total phosphorus doesn't match up as well with, say, USGS monitoring, where they're doing all the TP, but we do have continuous turbidity for the entire time, every 30 minutes. So we've got TSS to turbidity relationships, phosphorus content of those sediments in the water column. We can do some back of the envelope, and one of my new students is working on that turbidity piece and the phosphorus that might be associated with that. So it's a very good point, though.
Okay. All right. The question, another one here, and folks, then after that, I can open it up for audience members if you want to unmute, but to what depth were you sampling for organic matter in the soils?
We did the same, 0 to 5 and 5 to 20 centimeters. Yeah. And the very similar trends when I only showed the 0 to 5, but very similar 5 to 20.
And Shannon put in the chat box about another paper that they have in review that does look at SRP concentrations and what they found. Okay, Dr. Crumpton raised his hand. Great, we can use that feature. So then I can call on you. Thank you, Bill Crumpton.
Yeah, so hi, Jennifer. It's good to see you again. Could you bring back up the slide where you were looking at the watershed scale comparisons for the nitrogen and phosphorus export?
Let me find. Okay.
Maybe there were the bar charts showing the.
Yeah. I think.
Yeah.
Sorry, I'm not that. This one.
That looks.
Oops! Oh my gosh! I'll just page up. Are you seeing it?
Yep. Hold on. That looks. Yeah. This looks like it.
Okay.
So, so when you express the averages here, the thing that I have some concern about is on the nitrate, for example, so in 3 out of, or in, let's see, 1, 2, 3, 4, 5 out of 8 years, there's a reduction in nitrate yield. But there was an increase in the nitrate yield in the wettest years. If you look at that entire period of record, it looks to me as though the cover crops will not have reduced nitrate yield at all over that combined period of record, because during wet periods, the cover crop sites exported more nitrate than the non-cover crop. The same thing.
No, so we meant. Yeah. So this is just but this is the whole watershed. So this is just at the outlet. So it's just in the Kirkpatrick, we only have 1 year, 2015 to the comparison. And then in Chateau, we have from 2008 to 2013, which is why there's an error bar there. So out of those 8 watershed years where we had cover crop, exactly as you said, 5 out of 8, we saw reductions. And here was the average of those reductions when it reduced. And then in 3 out of 8, it was big water years with higher than those past years as reference. So there's no side-to-side comparison because we don't have control.
But if you're using these data to suggest that cover crops, because in 5 out of 8 years had a reduction in nitrate, you also have to use these same data to suggest that, well, overall, there was no reduction. So I know it's not a paired watershed comparison with one with nitrate or one with cover crops and one without, but if using the pre and post and arguing that post cover crops, 5 out of 8 years, you saw a reduction. But overall, in those 4 years post cover crops, it appears there was no reduction. And I ask that because in some work Matt and I are doing at the watershed scale, with better than 3 years of pre-planting paired watershed and 4 catchments, and then another 3 years post cover crops. And the cover crop percentages we're seeing there were, I think, over 50% in each of those catchments. And we can't see any effect of the cover crop implementation at that scale.
Yep. Yeah, I get your point, but it all depends on how you look at it. If we had done the study in 2016 only, I would tell you that it was a 30% reduction in Chateau and a 65% reduction in Kirk. If I did it in 2 years, then it's, this year, you know what I mean? It's all driven by the water year. And so all my point is with this is that water overwhelms this. It doesn't mean that cover crops aren't doing something. It means that there's this overriding driver of precipitation and runoff that we have to sort of put into context. And so the years we didn't see reductions compared to the pre-treatment years are the years that we had the most water coming off the watershed. And that's all I can really, I guess your point's well taken.
We do have, there's another question in the chat box, and then we'll open it up again for other questions. And given the impact of high rainfall rates, are some of these edge of field practices, I would say, the question is, are wetlands, and I'd say edge of field practices even more important to implement with cover crops?
Absolutely. I mean, I guess I would say that it's, and I wouldn't just stop at the edge of the field. I'd go right into the waterway as well. That this is not that cover crops are the only way to do this. It's that every square inch of ground, be it wet or dry, should have something on it that makes it more like a sponge for nutrients as opposed to a conduit. And so the dream would be cover crops on all the surrounding acres, then some sort of edge of field polisher like a constructed wetland or a bioreactor. And then if it gets into the waterway, something like constructed floodplains like the 2-stage ditch that makes it better than a pipe, So I think the whole thing is just sort of a suite of management choices to make it more like a sponge.
Yeah, protecting that land all the way from the field to where it's in the water body. Yeah. Are there other questions from attendees today? You can either raise your hand or unmute yourself and talk.
Well, for the end of the semester, pretty engaged. You have a lot of people. I'm honored and so happy to have been able to be here.
Oh, okay. Yeah. So at the $45 per acre of cover crop each year, have you calculated the cost per pound reduction at all?
Yeah, we haven't, but we're working on it. Well, we're working actually with John Tyndall. So so John is engaged and has been part of the project, and hopefully there'll be a sort of the economic valuation piece of it will be forthcoming.
Okay, that's great to hear, an Iowa State connection on this study. So maybe
totally.
Uniformity in calculating these costs across the Corn Belt then. Well, I just, if we don't have any other questions, I just want to thank Dr. Tank for an engaging presentation today. What a great way to wrap up our seminar series, and I really thank people for being on. I know as we talked before, for some the semester is over, and so some people may be, in the summer session or summer period, and thank Jennifer for being willing to talk to us after her semester is done as well. I assume she's on top of things and has grades all submitted and everything for that. Oh, maybe not. Okay, don't talk about it.
No, Matt, they do. They don't let you go to graduation without it. Okay. Someone will come knocking on your door.
Yeah. Okay. So thank you so much, and be looking for announcements for seminar series next fall, and also feel free to go back and watch some of the previous seminars that we've had. They're archived on the Iowa Nutrient Research Center website. So thanks again, Jennifer.
Thanks so much for having me.